Okay, all right. Well, I think we just get started. So just to be sure, I don't see her on my participant list, but Amy, you are not here, correct? Okay, we will go ahead and just start with number 2 and still work our way down the list. And then hopefully Amy will join at some point. And if she does, then we will just let her go at the end. And so what we'll do is each researcher will briefly kind of talk about the research that they do in their lab. And then I'll open some breakout rooms, which will all be numbered just as you see on this slide. And then you can join some of the researchers in breakout rooms to ask more questions about their research and hear more. Yeah, so as you're hearing people talk, if you think that they might be someone that you would like to speak with more, just kind of jot down their number. But we will start then. John, is it you that's gonna talk for your lab? Awesome. Okay, well, we'll go ahead and start with you then, John.
Great.
And I will stop sharing in case you want to share anything.
Cool. Jules, I'll pull up the slides.
Okay, that's good.
Right. So Jules and I are part of Dr. Hopfinger's cognitive attention lab. Here's kind of the things that we cover. So, Jules, if you want to start off.
Yeah, so basically, just to give a brief overview in case you haven't studied attention yet, visual attention is basically allocated according to priority, meaning that our brains rank the available visual stimuli in front of us to decide what we're actually going to pay attention to. So there's a lot of debates in the world of attention right now, especially something around like whether attention is mostly guided by what pops out at us or what we mean to look at. There's also other areas of attention research, like attention networks, which John is going to talk about real quick.
Sure. A lot of the stuff we work on in terms of attention networks is kind of based off of the work of Michael Posner, and that kind of dates back to the 80s and the 90s. So very kind of well-developed field. The main gist of attentional networks is that there's proposed to be 3 kind of distinct but still codependent networks, being alerting. So that's achieving and maintaining kind of an alert state where you're on the lookout for something that's coming up in your environment. There's orienting, in which you kind of selectively allocate your attention, as Jules has pointed out, to some spatial location around you that you deem more relevant. And finally, there's executive control, in which if there's conflicting stimuli, your brain kind of ranks them in order of salience to you, and you can pick and choose what you're paying attention to.
So for the methods that we use in our lab, we use a lot of behavioral studies, which recently during COVID has meant a lot of online studies. And I'll talk a little bit more about that in a second. We also use EEG and ERP studies, which also we'll talk about in a second, and neurostimulation as well. Finally, we use fMRI sometimes, which we're not going to go into more detail on later, so I'll spend a little bit longer on right now. Basically, this is a way of essentially getting a look at where in the brain is active when you're doing a certain task. So, for example, say I give you an attention task to do. You're looking at a screen and paying attention to one thing or the other while we're scanning you. It's basically going to tell us what parts of your brain are using more energy, and thus we figure more active while you're doing that. So John, if you'll go to the next slide. Yeah, thanks. So behavioral research is instead of using sort of a more biological approach, like actually scanning the brain and such, behavioral research is looking at things like response time. So for example, I'll talk a little bit about what one of my studies looks like. So John, if you'll click once more. So in this study here, what you're looking at is one of my stimuli, where basically I'm looking at whether or not that singleton distractor, the red circle, is actually distracting the participants, or if they can kind of filter it out. So on each trial, they're looking for the diamonds. They're going to respond with whether the number is greater or less than 5. So that's kind of what this looks like. And then for the results from this experiment, John, if you click once more, basically what we find is that the distractor being present actually means that you respond more quickly, which is super interesting, because then that means that we're just so good at suppressing distracting things. Okay, John, so now John is going to talk a bit, actually I'm going to talk a bit about EEG. So basically an EEG is a test that detects electrical activity in your brain. It uses these small metal discs called electrodes that are attached to your scalp. And what it does is it picks up the electrical impulses that are how your brain cells communicate between each other. They're active all the time, even when you're asleep. And so what this looks like when we're actually considering the data is we look for, we take trial, like tons and tons of trials, and we essentially average the waveforms that you're seeing on the screen right now, so until we get one sort of basically an average waveform for a person's response to many, many trials. And so, for example, one thing that we might look for is a specific negative, down pointing instance, where you can see some little waves in here at a specific point where the line is going to go down a bit. That's going to show us when you're actually paying attention to something and when you're suppressing a distractor, like I was just talking about in the previous experiment. It's super cool. Okay, so now I'm going to give it back over to John.
Okay, I'm going to be talking a bit about neurostimulation. This is the main experiment that I'm working under Hannah, who's one of our other grad students. She couldn't make it to this meeting today, unfortunately, but I hope I do her work justice. So neurostimulation is kind of a technique in which we place rubber pads, we stick them via electrolyte paste, and we make proper contact with your scalp. And the thought is that by applying current kind of through those rubber pads and across your scalp, we can actually modulate the underlying brain frequencies in the tissue that's underneath. And so Dr. Hopfinger, along with one of our previous grad students, John Parsons, had done a study in 2016 where they looked at the effect of neurostimulation on attention, and they'd actually found that certain different types of stimulation frequencies had helped either to improve disengaging from cues that weren't as significant to you, or that different types of stimulation had actually increased the ability for outside stimuli to kind of grab your attention away from your task. The current study that we're working on is an in-person one. We're still in progress, but we're about to wrap things up with participants. But Hannah and I have been looking at, again, the effects of stimulation on the 3 networks of attention that I've talked about earlier. And we're expecting that, and we're predicting that theta stimulation will increase kind of how closely you can pay attention to the task, and that gamma stimulation will increase the orienting effect. And pertaining to our undergrad peers with us here, I'm a senior myself, not a grad student. So getting involved in research is definitely something that's very doable at UNC. If you're looking on a volunteer basis, you can definitely look to ask any professors that you have class with. If they cover any research in class, you can just go to their office hours, knock on their door, say, hey, I'm really interested. And definitely don't feel like, oh, I don't know a lot about the field. Let me take more classes before I get involved. You can definitely get involved earlier. I kind of made that mistake earlier in my career as well. And I'm kind of kicking myself for it. But at UNC, we also have independent research for credit. And you can have that approved by the department. do reach out to professors a bit earlier, because you do have to kind of interact with them and make sure that you set up a proper mentorship. But you can go in with your own research questions, or as in my case, I kind of just tagged on to Hannah's work, and you can assist with works in progress. All right. That's all we had for our presentation.
Awesome. Thank you so much. Yes, and I was going to point out that John is an undergrad also. So if you're wondering how to get involved, that might be a good breakout room to go ask him more about what he actually did to get involved. Okay, so next up, we have Dr. Mark Shen is going to talk about his research.
All right, hold 1 second. Sorry, we tested this and now it's not working. Okay, here we go. All right, well, thank you so much. And it's a real delight to be talking to the undergrads. My lab and I, our undergrads and RAs, we actually discussed who would be the best person to give this presentation, and they decided it would be me. But they helped me put together these talks, and then I sent them out to lunch because we have a really fun lab, and they've it's been a long semester, so they're all out to lunch, and they put me in charge of doing this. So, but I'm really delighted to do so. I an assistant professor at UNC. My research lab is affiliated with the Neuroscience Center. I teach the autism unit to actually the PhD students in neuroscience. So it's particularly, I think, gratifying for me to connect with undergrads because a lot of the important work that we do actually starts with undergrads who come to us with really exciting ideas and a lot of passion for what we do. And I also represent the Department of Psychiatry, which is at the School of Medicine. So a lot of the work we do are with children with psychiatric challenges, and also the Carolina Institute for Developmental Disabilities, which is a both clinical and research center, part of the School of Medicine. And as you'll see at the very end, offers a lot of opportunities, volunteer and otherwise, for undergrads, for people to get involved with. And I have a couple of examples of that at the end of the talk. So our research studies babies, particularly studying the brain development of babies using MRI. And the goal of our research is to identify the earliest brain markers of autism. And so I thought maybe I would start with telling you what is motivating our research. So one of the important challenges that's facing our field as far as identifying autism earlier is a set of challenges that we've been trying to address. So first is that the current way that autism is diagnosed is based upon observable behaviors. That is the criteria for autism, that a clinician needs to be able to observe certain behaviors or certain symptoms. But that's a challenge because those behavioral symptoms don't actually emerge until at least 1 to 2 years of age. In other words, they're not evident to a pediatrician or to a clinician until that child is at least 1 or 2 years of age. The other aspect that makes this challenging is that there's a lot of individual variability in how individuals with autism present. And there's kind of a saying in the field that if you've met one individual with autism, then that means you've met one individual with autism. And I have family members who are on the spectrum. I've worked clinically in the community and in the school district with individuals, and that certainly is the case. And the way we're kind of diagnosing autism is kind of lumping all those individuals together. And so the result of all of these different challenges is that autism is not typically diagnosed until 3 to 4 years of age. And so even as the prevalence has become, quite common, the current estimates are 1 in 54 children in the United States will be diagnosed with autism. The age of diagnosis, the point at which we can actually diagnose these individuals has really not decreased in the last 10 years. It's still kind of hovering at 3 to 4 years of age. And, why is that important? It is because, like almost any other medical challenge or subtype, the earlier you can intervene and provide supports and services to that individual, the better. And so in autism, that's certainly the case, that early intervention or providing early behavioral supports or services to that individual is really, really important. The earlier, the better. And if you can provide those services earlier, then it can have really important beneficial long-term outcomes. It improves the long-term outcomes of that child. It improves quality of life, and the family in general support just improves. And so what has motivated our research are essentially like two questions, which is that, are there identifiable markers in infancy, so prior to 1 to 2 years of age, that can help us perhaps detect the likelihood that a child will go on to develop autism and do that earlier? And if that's the case, then that will allow us the ability to begin supports and services earlier in life and improve long-term outcomes. And then second, can we sort of leverage this individual variability that we see in individuals with autism into biological subtypes? So instead of sort of lumping all of autism into sort of one behavioral criteria, are there biological signs that we can sort of parse people into so that we can hopefully develop more targeted or more individualized interventions or treatments for those individuals? So how do we do this? If autism is not diagnosed until 3 to 4 years of age, how do we study babies prior to diagnosis? And the way we do this is what a study design that's called the infant sibling study design. And essentially what this does is it leverages the fact that the recurrence rate of autism spectrum disorder in siblings is about 1 in 5. So in other words, if you have an older sibling who already is diagnosed with autism and you're born into that family, you have a 1 in 5 likelihood of developing autism yourself. So compared to the general population, which I told you was about 1 in 54, it's about a 10-fold increased likelihood if you have an older family member, older sibling with autism. So then what we do is we follow hundreds of these babies who have an older sibling with autism, and we follow these siblings from birth. And we do longitudinal or repeated MRI scans and behavioral assessments in these babies across the whole first 2 years of life until the age that we can diagnose them reliably around age 2. And then we can look at all of their brain and behavioral data from birth and parse out which are the behavioral and brain markers that identified babies who ended up developing autism and those who did not. And so far, we've studied about 800 babies. UNC is the lead site for a multi-institution study that's been running for about 15 years now. It's a collaboration with 5 universities from across the United States, some of the top schools of medicine. And like I said, they travel to Chapel Hill and these other 4 universities for these repeated MRI scans. And our ultimate goal, as I said, is to search for early markers of autism before diagnosis is currently possible. So this is a couple images, little pictures taken of some of our babies. This is a 6-year-old baby who was in our study. We have little, if you've ever had an MRI, it can be quite loud, and we don't want to use anesthesia or sedation for these babies. So we are actually doing MRI scans during natural sleep. And the way we do that is, first of all, we protect their ears really well. We have little baby noise canceling earplugs, little baby noise canceling headphones. And then we also play the sounds of the MRI for them while they're sleeping before they travel to Chapel Hill. And the auditory system actually gets used to those sounds, so that when we put them into the MRI scanner, which is down here, we have about a 95% success rate of these babies sleeping through the MRI scan, so we can get detailed pictures of their brain. The other thing that we do is we also decorate the MRI machine. So we have several different themes. This one is our astronaut theme. So we decorate like a space rocket, and that way, babies feel a little bit more comfortable because it's not this really, medically intimidating environment. It kind of looks like sort of a play area. And I'll just give you a little bit of a summary of what we have reported to date through this research. So here on the left-hand side are 5 different aspects of brain development that we have detected are brain markers that are evident with MRI in the first year of life. So this is before behavioral symptoms are even showing in these babies. We found that they have an excessive amount of cerebral spinal fluid that surrounds their brain, that their brain is growing too large, or there's brain enlargement in the babies who would go on to develop autism, that there are different connections of their brain, both white matter and functional connections are altered in the first year of life. That's by 6 months of age, so 2 years prior to them being diagnosed. And all of these brain markers, as we've reported, are detectable with MRI prior to them showing the diagnostic behavioral symptoms of autism in the second year of life. And these symptoms here are what we currently use to diagnose. But what we're seeing is that these brain changes are occurring well before they're showing symptoms. And each of these 5 brain markers are able to actually predict either which babies will go on to be diagnosed with autism 2 years later, or how severe their symptoms are going to be, or associated with what some specific symptom profiles that are individualized for each child. So this is what is sort of driving our research is this idea that we can move towards looking at more biological neuroscience changes in the baby's brains and do it non-invasively with MRI, and that will help us be able to detect autism earlier and hopefully provide sort of better services and earlier interventions to improve quality of life in these children. So this is typically where I would end and just acknowledge sort of all of the families and children who participate in our studies. I have a really wonderful lab of undergrads and research assistants, doctoral students and postdocs. But actually what I wanted to focus on was just to highlight the stories of 2 undergrads that work with our group who are now kind of moving on and have worked with our research lab and now going to grad school. So Julia Gross came to me when she was a sophomore as an undergrad major in psychology. She just emailed me sort of out of the blue, and she had a particular interest in brain development. Her undergraduate project, which she led to a co-authored paper on infant brain development in autism. She was such a good undergrad research assistant that I hired her after she graduated. And now she is working full-time in my lab as a study coordinator and my lab manager. She's collaborated with faculty at several different universities, and she's now presenting at an international conference in May. And she's starting a new brain imaging study of infants and also helping us with some clinical trials to actually delivering new treatments that have never been delivered before, and that's at UNC. Here's her email address in case you want to reach out to her. Alapika Jatkar just graduated a year ago in May of 2021. Well, not even a year ago, 6 months ago. As an undergrad, her work led to a first authored publication on the early behavioral markers of autism. Again, was so impressive in her undergraduate work that after she graduated, was hired full-time as a study coordinator with Dr. Becca Krasinski, who's also at the CIDD with me. And we're very proud of Alapica because she just got into her first choice of her doctorate program, which is a doctorate in optometry that she'll be starting next fall. And Alapica was kind enough to record a little video that she posted on the Sakai site that tells her story. And both Julia and Alapica would be happy to talk to any of you guys if you guys are interested in our research. And of course, there'll be a breakout room where you can talk to me. So again, thank you so much. We're hoping that you're interested in our work, and please reach out to us if this sounds exciting to you and there's something you want to do while working with us.
Great, thank you very much. All right, I'm just gonna move on. Make sure we have plenty of time to have some time for questions. Next up is Mallory Feldman. Thank you, Mallory.
Yeah, so I'll be pretty quick. Unfortunately, I don't have slides, but so I'm a third-year graduate student. I work in the Carolina Affective Science Lab with Dr. Kristen Lindquist, and we're in the social area of the Department of Psychology and Neuroscience. And our lab is super interdisciplinary. We use methods from social cognition, physiology, and neuroscience to understand how people experience emotions in their own bodies and perceive emotions in others. And a lot of our work looks at kind of how people's relationship with their bodies, with the kind of sociocultural context that they find themselves in, impact how they feel in their health over time. I'll just note that we have kind of 3 big studies that are running right now that we'll be recruiting RAs for soon. And so I'll give a 1-sentence description of both of those, and then I'll probably just wrap up. And I am really excited to talk to y'all in breakout rooms. So the first study that I'll mention is called Super Teen. And for this project, Dr. Lindquist is collaborating with Dr. Eva Telzer, who's in the developmental psychology area here. And for this project, we are following kids as they enter and leave middle school. So it's a longitudinal project. And we're looking at the kind of development of different socio-emotional processes as they pertain to adolescent risk-taking. That project's super exciting because we get to work with young kids. We follow them for a really long time. We get a ton of data from them. A subset of them are scanned. And so it's a huge team of folks working on that project, and many different interests. So the data is really, really rich. The second project that I'll mention is the one that I'm working on primarily, so I can speak the most about, which is the lifespan study. So we're recruiting folks from the community, ages 18 to 80, and we're looking at how kind of changes in the peripheral nervous system relate to changes in mental experiences of feeling states like emotion. And then the last one I'll mention is a project called SANI. I wish I could tell you what that acronym stands for, but in full honesty, I don't remember. It's a brand new project that's looking at how experiences of positive emotions change connectivity patterns in the brain. And so for that, we're collaborating with another researcher here in the social program named Dr. Barbara Fredrickson, who's very well known for studying positive emotions in particular. So those are just 3 studies that we have kind of on the horizon that we're kind of like actively recruiting for. But in general, our work is quite broad in its scope. Last thing I'll note is that we get a lot of folks who are interested in pre-med or who are trying to adjudicate between pre-med and a career in psychology, just because so much of our work involves measurement of peripheral physiology in the brain. We're increasingly doing that at the same time. So getting these kind of really scoping measures of peripheral physiology at the same time as we're collecting brain data. And so a lot of folks who are interested in kind of walking that line, figuring out if research in psychology is something that they'd be interested in, given kind of a priori interests in biology or anatomy, come to work with us. And, you know, we love having all kinds of folks come and work with us, but we've found that those folks in particular tend to find like a happy home with us. So please come chat with me or email me. I think our information's on Sakai, but I'll stop there and pass the mic.
Thanks so much. All right, next up we have Sequoia Ashburn.
Hi, everybody. I am going to share my screen. Although PowerPoint's not showing up. 1 second. There we go. So I am a postdoctoral fellow in Dr. Cohen's lab in Psych and Neuro department. Most of my talk is on my own research, but also with how my research relates to the lab overall. So my main focus is on the cerebellum, which is this brain region down here. Most people think of it as like a motor function region, helps you with balance and coordination, but there's a wide range of literature showing that it's also involved with higher cognitive functions. So my interest is understanding how it's related to higher cognitive functions. So that's things such as executive function, working memory, most things that you can think of, and how it also relates to neurodevelopmental disorders, since many of those disorders have deficits in either higher cognitive functions or in the cerebellum. And for the lab, this overlaps because there are quite a few studies that show abnormal cerebellar findings for ADHD. So I'm interested in how the cerebellum is related to ADHD, specifically at the moment. I have also studied like dyslexia and other neurodevelopmental disorders before. So ADHD, known as attention deficit hyperactivity disorder, is the most common neurodevelopmental disorder in children. And roughly 14% of kids have ADHD, but it's still largely thought to be underdiagnosed. And one of the reasons why it is a disorder of interest is because a lot of the characteristics like inattention, impulsivity, hyperactivity, they're all things that impact our learning, our academic ability, our social abilities. So it is a disorder that can have a large impact on someone's life. And all of these can manifest in many different ways as shown over here, which can also lead to difficulties with diagnoses. So our lab focuses a lot on functional connectivity. So functional connectivity is thinking of how two brain regions fluctuate with one another. So we use MRI and we can get a measure of how particular brain regions fluctuate over time. And if those two brain regions are fluctuating similarly or in sync with one another, then that's thought to be called connectivity. They're connected functionally to one another. The lab that I'm in right now with Dr. Cohen does something a little more, well, let's say sophisticated, than just looking at two regions. We look at a plethora of regions. So we don't just look at the connection between two regions, but between multiple and how they can operate as a network. And there are a bunch of measures under graph theory where you can describe what networks are like if you're looking within connections, or if you're looking across networks, or even if you're looking at a particular point within a network and how it's relating to the rest of the network. So all of these measures can help us understand a bit more what one particular region is doing in a given network. And for my purposes, that's great because I can understand a bit more of how the cerebellum is related to other brain regions that we know are implicated in ADHD. And one of the reasons why I am focusing on the cerebellum again is because it is a highly connected structure. So this paper did a really cool experiment where they used some fancy statistics and mapped out all the different, or 7 networks, not all of them, but they mapped out 7 networks on cortex. And then they said, okay, we found these networks in cortex, but where are they in the cerebellum? So they did a similar set of fancy statistics, and where they're showing this black dot moving around is in cortex, but they're showing on the bottom where the corresponding regions are in the cerebellum. So they were able to show that all the networks represented in cortex also exist in the cerebellum. That's great for my research purposes because that means that the networks that I'm interested in for ADHD also appear in the cerebellum, and that it's also worth looking at and continuing with this research project. So again, my personal overarching question is how the cerebellum is functionally related to cortical networks implicated in ADHD. The lab itself uses the graph theory metrics on a range of questions related to ADHD. So undergraduates in our lab, they can assist in a bunch of ways. They can help with the actual data collection, which we are in the middle of right now. So getting kids in the scanner and getting the different types of scans that we need. Assistants have also helped out with some of the behavioral testing, so testing for IQ and other ADHD criteria. And then many of our assistants actually help out with the data analyses. So you do get your hands involved with that. And that could be at the level of behavioral, so looking at that neuropsych data. It could be looking at functional activation, which regions are active, or even looking at the connectivity that I mentioned, the network level connectivity, and how particular regions or particular networks interact with ADHD. And I always have a question slides out of habit, but feel free to follow me on Twitter. It's all science, or LinkedIn if you happen to use that.
Yes, and if you have questions, definitely join Sequoia's breakout room and chat with her a little bit about her research. Okay, we have 2 more people to get through, and then we'll open up some breakout rooms. So the next person that we're going to hear from is Klein.
All right, so I'm not great at sharing my screen. If this is wrong, Vicky, please unmute and let me know. Hopefully it's showing the right one. Great, thanks. So my name's Claire, and I'm a 2nd-year graduate student in the clinical psychology program. And today I'll be presenting on research that sits within the UNC Teach Autism program. So first, a short background. Teach is a network of 7 outpatient clinics across the state that serves folks on the autism spectrum. And in addition to our clinics, we have the CLLC that I'll talk about at the end, and we offer employment services and professional trainings around the world, and we do research. So Teach's mission statement is to create and disseminate community-based services, training programs, and research to enhance the quality of life of individuals with autism spectrum disorder and their families. And then within Teach, we have 3 researchers who all specialize in adolescent and adult outcome within autism. So you'll hear from Dr. Maddox later in the week, but then Dr. Tomaszewski also works in our lab, whose main focus right now is employment for individuals with developmental disabilities. But today I'll be talking about a project that Dr. Tomaszewski and Dr. Klinger collaborate on. So I sit within Dr. Klinger's lab, and she's the executive director of Teach, and her work is focused on autism across the lifespan, especially autism in adulthood and the transition to adulthood for autistic young adults. And our major project that has opportunities for students is our current randomized control trial, which is examining the Teach school transition to employment and post-secondary education, or the T-STEP program. So the T-STEP is an intervention for 16 to 21-year-olds with autism spectrum disorder that was developed to support the transition to employment and post-secondary education. And then in our RCT, students are either assigned to the T-STEP or to individual counseling. And the T-STEP is a program that covers many different aspects of the transition to adulthood, and it covers areas specifically that are important for success in college and employment. And these are also areas where individuals with autism may encounter challenges. And then in the individual counseling portion, they attend counseling meetings for career or job exploration counseling. And then if you're a research assistant with us on the project, you would be doing fidelity to the intervention coding. So you would get to watch the intervention sessions and help us code them. And you would be helping with behavioral coding. And then if you're interested in direct assessment, we may also have training opportunities with that. So here's an example of a measure you would be coding. It's the transition readiness and employability evaluation. It's our main outcome measure. And the light blue bars are showing improvement post-intervention. So you would be helping us score folks on executive function, social skills, and emotion regulation. So if you're interested in helping with research, These are some of the opportunities that would be available, and we can talk more in the breakout room. And then if you're interested in a paid position working directly with adults on the autism spectrum, we have some opportunities available at the Carolina Living and Learning Center, which is an integrated vocational and residential program for adults with autism. And we can talk more about that in the breakout room as well.
Thank you so much. I love that all of you are really giving like specific details about what undergrads do in your labs, because I get that question all the time and I'm like, it varies so much by lab. You kind of have to just talk to the lab. All right, we have one last person. So we're gonna loop back around, and Amy Careless, I'm not sure if I'm pronouncing your name correctly, is that right?
That's good enough. Hi, okay. Share. Okay, so I'll be presenting on some information about the Circle Lab. So hi, I'm Amy. I'm a post-bac research assistant in the lab. So I just graduated in May. And yeah, so we are the Circle Lab. So we study early life experiences and brain development, which I'll go a bit into detail about. And we are in the clinical and the developmental part of the psychology and neuroscience department. So this is our wonderful lab. We have a ton of lab members, and we are so lucky to have two main PIs. So Dr. Margaret Sheridan and Dr. Adam Miller are our two principal investigators. And then as you can see, we have a ton of research assistants, all post-bac, a couple grad students and postdocs, and a lot of undergraduates. So there's a lot of mentoring in our lab. There's a lot of opportunities to meet really cool scientists and learn about all the cool things that they're doing. So like I mentioned, we have a lot of different studies going on in our lab, but they all sort of converge on this idea of studying the impact of different life experiences on behavior and the developing brain. So in this lab, we have an interesting way of looking at adversity. This is sort of novel and what Dr. Sheridan is working on. And it really breaks apart these early life negative experiences into experiences of deprivation, which are things like neglect and institutionalization, and experiences of threat, which are things like abuse and exposure to community violence. So by parsing these things apart, we see that they have really differential impacts on things like brain development and social emotional development, which is really what we're trying to hone in on. So how do we study adversity in the brain? We have a couple of different techniques to do this. So the first one is interviews and questionnaires. And we work primarily with children and adolescents. So like in our children's studies, we have children versions of these questionnaires and then adolescent versions as well. We also have behavioral tasks. So we look at questions about executive functioning and emotional reactivity, and those are all done with behavioral tasks. And then finally, we do brain imaging. So we have both EEG, which was explained very nicely earlier, so I won't go too deeply into that. And then we also do fMRI and MRI analysis. There's different hypotheses about how early life adversity sort of differentially impacts brain development. And so as we pull all these things together, we can get a really cohesive picture of what that looks like. So something that we really wanted to emphasize in this presentation is what can the Circle Lab do for me? And so these are really good positions for individuals who are interested in psychology graduate school or medical school or something like that, or neuroscience as well. Also, if you don't know what you would like to do, that's also totally fine. Like I said earlier, we have a really big mentoring structure in our lab, and we have so many different projects going on that you can get exposure to different things. Like if you really want to hit the EEG side of it versus if you're really only interested in like the questionnaires, there's definitely a lot of room to grow and to wiggle. So these are the positions we have open. We have some federal work study and Carolina work study positions open. And we have two different kinds of jobs that you might be doing. So the first job is you would be performing hen edits of MRI data of cortical structure. So this is sort of a really unique position for undergrads where you can learn something called free surfer, which if you're interested in doing graduate school in psychology or neuroscience is a translatable skill. So it's a good thing to have. And then the other task that you might be doing is coding videos of parents interacting with their children. So this is another common technique in developmental psychology, learning how to code videos using like a standardized method and with a team. And because they're paid, we do have a minimum of 10 hours and then up to 20, just based on the work study rules. We do have a diversity, equity, and inclusion statement. We just want to note that we're really interested in sort of branching out the diversity of our lab. We do study sort of underrepresented populations, and so we think it's really important to have a team that's really in tune with those nuances and the possibility of just making sure that voices of our participants are heard. And so we want to emphasize that. This is just a little bit of the application process. And then I couldn't have any undergraduates with me today, but maybe in the breakout room, I can read you their testimonials. They're very excited and they're very, I love them dearly. So I can read you more about that. But we also have a couple more information sessions coming up this week. If you want to learn more, there'll be postdocs and grad students at those. You don't need to memorize the Zoom links. They're on our website if you're interested. And that's what I have. Keep it nice and short.
Awesome. Thank you so much. Okay, I am just going to pull up the slide from the beginning there. So what I'll do now is I'm going to open, okay, so y'all can see that slide, right? That has the numbers. It's not presented, but that's okay. Okay. So yes, I'll open 6 breakout rooms that will be numbered 1 to 6. And so if you are one of the presenters, if you can go to your correct breakout room, and then students, you can join them in those breakout rooms as well. I'll probably try to keep it to 5 or 7 minutes so you have an opportunity to go to at least a second breakout room. I know some of our presenters need to leave